Why do pathogens modify surface antigens to escape immunity? Imagine a game where the rules keep changing just as you think you understand them. That's what pathogens do with their surface antigens to stay ahead of our immune system. Surface antigens are molecules on the outside of bacteria, viruses, and parasites. They act like flags that tell the immune system, "Hey, I am foreign, attack me." When the immune system spots these flags, it responds by making antibodies and activating immune cells to destroy the invader. But pathogens don't want to be caught. They change their surface antigens to hide from immune detection. It's like they're constantly changing their disguises so the immune system can't recognize them. This process is called antigenic variation. It makes it hard for the immune system to remember and recognize the pathogen. When antigens change, the immune system's memory cells, which remember previous infections, become less effective. This allows the pathogen to stick around longer and cause repeated or chronic infections. Pathogens have clever ways to modify their surface antigens. One way is through genetic mutation. They change the genes that code for their surface proteins, creating new versions that the immune system doesn't recognize. Some pathogens also recombine gene segments, mixing and matching to produce new antigen types. Another trick is phase variation. Some bacteria can turn certain surface proteins on and off, changing their appearance during infection. Then there's molecular mimicry, where pathogens produce antigens that look like the host's own molecules. This confuses the immune system and makes it harder to attack them effectively. Viruses like influenza mutate their surface proteins, such as hemagglutinin and neuraminidase, to escape neutralizing antibodies. Bacteria like Neisseria meningitidis change their surface structures to avoid antibody binding. Parasites like Trypanosoma brucei regularly switch their variant surface glycoproteins to stay hidden from immune cells. The immune system fights back in a few ways. Some antibodies can recognize conserved parts of antigens that don't change much. T cells can detect infected cells by recognizing pathogen peptides presented on their surface, even if the surface antigens are different. Natural killer cells can also identify infected or altered cells without needing specific antigens. But antigenic variation remains a big challenge for vaccine development. Vaccines usually target specific, stable antigens. When pathogens change these targets, vaccines become less effective, sometimes needing updates or new designs to keep up with the changing antigens. So, pathogens modify their surface antigens mainly to avoid being recognized and destroyed by the immune system. This ability to change makes it harder for the immune system to remember and fight them off, allowing these microbes to survive longer inside the host. Understanding how they do this helps scientists develop better vaccines and treatments that can keep up with these clever tricks.